The Autism Research Institute relies on the generosity of donors like you to make this webcast possible. If you enjoy this presentation, please consider making a donation. Thank you.
Before we get started, I'd like to introduce our speaker. William Parker is an associate professor of surgery at Duke University School of Medicine. Dr. Parker's research interests include the promicrobial aspects of the immune system and investigating the immunological differences between laboratory-raised and wild-raised animals as a probe for differences between humans with and without modern medical care. This comprehensive approach has turned up unexpected and dramatic differences between the unhygienic and hygienic populations. And now I'm going to turn it over to Dr. Parker.
Thank you very much. I'd like to thank the organizers for inviting me to speak with you today. So I don't have any conflicts of interest to report. I will be describing some commercial products, but I don't have any financial interest in any of those. Most of the work I'll be describing today and all of the work that we do in our animal models and our rodent models is done in collaboration with Stacy Bilbo. She's a professor here at Duke, very well respected in the scientific community, and is an expert in how the immune system and particularly inflammation affects the developing brain. So I need to give her due credit before I start this talk. When we talk about how the immune system affects the brain, I think it's important to understand that in Western culture, the immune system is very, it's basically messed up, for lack of a better phrase. We have a lot of problems in Western culture. The allergy rates are at 40%. Almost 8% of folks have some kind of autoimmune disease, depending on how you count issues with the thyroid. And I think the most shocking statistic right now is that 43% of our children are on some kind of medication for chronic conditions. Most of those conditions involve inflammation and immune dysfunction. And of course, lastly on this slide, most of you are aware of the increasing numbers of cognitive disorders that we have in today's culture. When discussing immune dysfunction, I think it's important to take a step back. This experiment that I'll show you on this slide is, I think, very illustrative of how our immune system works. So on the left, you're looking at a cassette that has gut epithelial cells on it. And these are epithelial cells that line our gut, the small intestine, large intestine. That we've grown them in a little cassette. It's about 10 centimeters by 10 centimeters square, about the same size that you see on your screen. But it looks blank, and the reason why it looks blank is because the epithelial cells are transparent, and the bacteria that we put in there didn't stick on the epithelial cells. They just washed right out. Now, in the human gut, you do have biofilms, and biofilm is a living film of bacteria that grows on the epithelial cells. On the right-hand side, you can see nice, thick bacterial biofilms that grew on those epithelial cells, and the only thing we did to make the difference here was we added an immune molecule, secretory IgA. Your body produces copious amounts of it and secretes it into the gut. So the idea here is that the immune system is actually supporting the growth of bacteria rather than being antagonistic to the growth of bacteria. And we think that in general the immune function is centered around maintaining symbiosis. It's not a sort of an antagonistic defense system trying to drive away all symbionts, but rather it's a system that you could think of as an interface with the environment. In the next slide, you're on the right-hand side, you're actually looking at a stained piece of normal human appendix. The bright, large, bright yellow spots, those are the nuclei of the actual appendix cells. And just to the left of that, the tiny little layer of, or the layer of tiny small spots, that's a biofilm. That's a bacterial biofilm. And this observation led us to figuring out what the human appendix does, which the human appendix is a bacterial safe house. It's a storage place for bacteria in the event that the system gets infected and needs to be flushed out, which is a diarrheal response. So we couldn't really understand what the appendix was doing until we understood that the immune system was supporting bacterial growth. So just in terms of basic biology and how the whole entire ecosystem of the human body works, it's important to understand that the immune system is trying to maintain function and maintain the symbiotic relationships that we need. So now we go back to this question. I think we can, if we understand what the immune system is trying to do, it's easier to address this question of why is our immune system so wacky. Now, about 30 years ago, this term hygiene hypothesis appeared on the scene. And the hygiene hypothesis, many of you might be aware of it, it's the idea that we're so clean that our immune system is not getting challenged. Well, it's a reasonable idea. It turns out it's not really about hygiene today. And I'll explain that in some more detail. I'm providing references for you. I know this is not for continuing medical education, but all the references will be in the slides if you want to look up any of the papers. All the references essentially for every single point that I'll be making today have been published or will soon be published, and the references are available. So the idea here is that it's not so much just being clean. In fact, today it's not being clean at all. It's more that we're missing biodiversity. And the biome is, simply put, it's all of the living species or all the life forms within the ecosystem of the human body. So in the ecosystem of the human body, we've lost a lot of biodiversity. We've lost a lot of the species. That is what is a primary factor that's destabilizing immune function. Now, biome depletion by itself, and I'll talk about what parts of the biome are being depleted, and we'll discuss the microbiome and so forth in a little bit, but first it's important to note that it's not just biome depletion that causes immune system malfunction in modern society. Biome depletion works hand in hand with other factors. I've listed, there's 4 major factors besides biome depletion. They're all listed on the slide. Vitamin D deficiency is primary because we work indoors. Chronic psychological stress, I'll talk about that in a little bit. We're just publishing a paper showing that relieving stress is a basic human need, like obtaining food and water. And of course, most of you are aware that the inflammatory diets and sedentary lifestyle that we have in Western culture are not good for immune function. The bottom line here is that you've got a lot of different things coming together to cause immune dysfunction. How does this affect autism? Well, it turns out that Kevin Becker, who is heads of one of the branches at the NIH, was the first to realize that autism is connected to biome depletion. And autism is almost certainly connected to those other factors that destabilize the immune system. So this is a mainstream concept. It's not a fringe concept, although unfortunately it's not been widely recognized by most researchers, I'm afraid. So this slide is a little bit complicated, but it's worth spending some time on. And the reason we need to spend some time on this slide is because a lot of people get confused. You know, is it genetics that causes autism? Is it something in the environment? Or, you know, what about all this inflammation? How does all this fit together? This slide, I think, does a very good job of putting that together. Now, it's a 3-hit paradigm. There's 3 things that come together to cause any kind of hyperimmune associated disease. And on the next slide, I'll get more specifically into autism, but first I think it's important to understand the general concept. So the general concept is that you have these 3 hits. The hit number 1 there is the immune hypersensitivity that I talked about on the previous slide. It's caused by biome depletion, inflammatory diets, vitamin D deficiency, chronic psychological stress, and sedentary lifestyle. So that's something that's inherent in our culture, unfortunately, and it interacts with things which some have been around for millennia. Some have been around since before the agricultural revolution 10,000 years ago. So, for example, the genetic predisposition that many of us have today for getting these diseases It's not because our genes are new. The genes have always been around. It's because it's interacting with other things. So the environmental stimuli are, in some cases, they're the same thing. They're ancient factors. The easiest example to explain is ragweed pollen and hay fever. Ragweed pollen has been around for millennia, but it didn't cause hay fever until the Industrial Revolution. It was first described, that is, hay fever was first described in the late 1800s, and people were struggling to figure out, what was causing it. So just because you identify a gene that's associated with the disease doesn't mean that the gene caused the disease. It just means it's associated with it. And I think it's important to look at how the three hits fit together, especially when we're trying to eliminate or treat a disease, because you can't change the genes so easily, and some of the environmental triggers cannot be easily changed. As I promised, we'll go to a more specific slide on autism. This is one we just recently published. It shows how, on the top left, the immune dysfunction interacts with inflammatory mediating factors. And these would be the triggers. And I've got acetaminophen on this list. Well, on the next slide, many of you know about acetaminophen and its possible role in the development of autism. Some of you don't. The next slide will go into some detail on that. But the idea here is that you've got a tremendous amount of oxidative stress. Glutathione is a central molecule in all this. In some cases of autism could be caused by this high degree of oxidative stress, which is associated with inflammation. It's a vicious cycle. Once the environment becomes toxic, the inflammation increases. And then, of course, finally, susceptible timing is very important for autism, as well as genetics. And then you've got your three hits there. So it's still a three-hit diagram. I'll show you another three-hit diagram later on. But the bottom line is that it's not just one thing. I think it's important for autism and understanding autism to know that there's so many things that can cause oxidative stress that you could easily, if you only test one thing at a time, it might be possible to prove that nothing causes autism. Because oxidative stress is a very common kind of insult that a number of environmental antigens and even our own bacteria, if you have leaky gut syndrome, something like that, can definitely cause increased oxidative stress. So, and the microbiome there obviously again comes into play. I promised you a slide on acetaminophen. I'm not going to spend much time on that. It's not something I do research on, but there's a lot of correlations between acetaminophen use. It hasn't been nailed down. My view on it is to presume it's guilty until it is proven innocent, just because the benefits of using acetaminophen versus other approaches are not that tremendous. And, of course, the advantages of avoiding autism, potentially avoiding autism, are tremendous. So back to the biome issue. And the reason why we're focusing on the biome is because it is, as best we can tell, it's the primary factor that is destabilizing immune function. Again, there are other factors. And it's also one of the main factors that we can manipulate to affect disease or to treat disease, and especially to prevent disease. So it's important when looking at the human biome to know that the microbiome is only a subset of that biome. So the microbiome, by definition, is all the microorganisms. There's other larger organisms that have always been with humans. Protozoans are a good example, and helminths, which are worms that live in the intestine. Those have always been with humans, and they're with all other vertebrates until the Industrial Revolution, and even more recently than that, until the mid-1900s, when toilets were universally installed throughout the Western world. That's essentially annihilated the helminths. So, and the microbiome clearly has been affected by Western culture. Bottom line is that the human biome is a complex part of our ecosystem, and it doesn't just contain the microbiome. All right, this is a sort of a view looking at the ecosystem of the human body as a three-legged stool. If you've got, it's about worms and germs interacting with the immune system, and the worms or the helminths affect things differently than the microbiome does. So they sort of fill two different niches. You can't, if you lose your microbiome, you can't replace those with helminths, and if you've lost your helminths and other complex organisms, you can't replace those with microbiome because, again, it's like a three-legged stool that is required for stability. If you lose one leg, the other two legs are going to have some problems compensating for the instability. The next three slides we'll look at are going to compare the two different parts of the biome that I've talked about in terms of how much does Western civilization destabilize those parts of the biome. So on the left here you see that the microbiome, it's a little bit beat up. Antibiotics certainly beat up the microbiome. Changes in birthing practices, probably use of infant formula beats up the microbiome. But the other biome components have essentially been annihilated. Here's some data in support of that. This is data from Jeffrey Gordon's lab. Jeffrey Gordon is a major player in the field of microbiome research, probably should be considered the father of microbiome research in the United States. He looked at the diversity, and here he's got a way of measuring, and we all use this, I use this myself, a way of looking at the number of species. You don't exactly measure the number of species because you can't get that much detail and still get a big picture of what's going on. So it's called an operational taxonomic unit. But it's a surrogate marker for the number of species present. What you see is that in the United States, we still have most of our species, when you compare that to the South American pre-industrial tribe and an African pre-industrial tribe. Now there's some decrease. There's some decrease in these operational taxonomic units. And an operational taxonomic unit is just a grouping of closely related species. So there's some decrease in the number of these species, but it's mostly related to diet. And furthermore, you get more variation within a population than you do between populations. Now, in the next slide, we're going to look at IgE levels in pre-industrial and post-industrial populations. And the reason to do that is because IgE levels in the serum are a very good marker for how much you're exposed to fauna or animal like protozoans or helminths. So as the IgE levels go up, it's just an indicator that we've been exposed to helminths or other kinds of more complicated organisms than what we normally think of as the microbiome. If you look at the very bottom green line on this chart and the top green line, you're looking at industrial and a pre-industrial population, respectively, that have dramatically different levels of IgE. In fact, the difference is more than tenfold. Now, if you look at those center two lines, where the bottom of those center two lines is U.S. maximum normal concentration, so that means that if you're in the United States, your levels don't go above that normally, and if they do go above that, it probably means you have some kind of raging allergy. But that maximum normal level is still below the level that we find in a pre-industrial population, even when we find no evidence of helminths or protozoans in their stool. So for pre-industrial populations, even when we don't see visible helminths, we can't find them, we can't find evidence of them, there's evidence in their serum biomarkers that they have been exposed to those, which would explain, based on a lot of other information we have, it would explain why those individuals don't have allergies and they don't have autoimmune diseases. The next slide is sort of took a little bit of liberties with Graham Rook's model, and Graham Rook is probably the father of the modern biome depletion theories that are available today. He divides the biome into three different groups depending on how Western society has affected our exposure to those groups. And again, this is modified based on his model. He uses a little bit different terminologies and a little bit different groupings, but it's essentially the same thing. And again, if looking at the top, we've mostly lost our helminths. They're essentially annihilated. We've diminished our contact with many of our good germs, especially some of the bacteria that would come from soil and get on our skin. But by and large, we still have most of those bacteria. In contrast, we've increased our contact with bad germs, and Graham Rook calls these crowd infections. So crowd infections are germs that we get because we live in very high population densities. We never would have had these things before 10,000 years ago when the agricultural revolution happened, because we were in small groups of hunter-gatherers. So importantly here, if you don't wash your hands, it probably won't help you improve your biome. In fact, you'll just get exposed, you'll get a lot more crowd infections, and you'll get exposed to a lot more inflammatory things. So it's not about washing our hands anymore. There's something else involved than just what we think of as hygiene. These are the kinds of things that are involved. The helminths that I work with in my laboratory, for example, can't survive if you have a refrigerator, if you have canned goods, if you have plastic containers, because the helminths that I work with require rodents as carriers, and rodents can't get access to your refrigerator. So again, it's not about hygiene. It is about all these things in modern society. Even shoes block the transmission of several different kinds of helminths that are important in human cultures that are pre-industrial. If you want general reading on this topic, I would suggest Moises Velasquez's book. It's a very good book, and you'll have the slides. It'll all be on YouTube, so you'll be able to access this information. I'm providing a lot of references here that you can access on the YouTube video. A lot of people ask me how the helminths work, how do they stabilize immune function, and we've done a lot of work on that. I do most of my research on immunology. It can put people to sleep. I think it's important to sort out exactly what's going on, but it's important from a long-term sort of visionary perspective, not necessarily in terms of fixing the problem right now. Right now, the main thing we need to say that it's very complex. So if you look on the left-hand side of your screen, you see that there's eggs that give rise to an adult worm, and then in the middle of the screen there's these spots that are on a little plate, and those spots each one of those spots represent a different protein, and the ones there, you can see there's a number of different spots. There's dozens of, not more spots there. Those spots that are circled are proteins that we know what they're doing. That's a two-dimensional gel there, the way that we separate out proteins that are in a complex mixture. And again, you can see there's a lot going on here that's affecting immune modulation. There's a couple of references there that you can take a look at if you are interested. These are a couple of more references that are not from my laboratory. Again, idea being this is very mainstream. People are aware that modern society is derailing immune function. Most people don't know that helminths can be very effective at treating disease. There's a paper published in 2007, almost 10 years ago now, showing that helminths will halt the progression of multiple sclerosis. Many people simply are not aware of this information. So what happened in this study was a group of scientists or clinicians in Argentina had about 500 patients that they were following with multiple sclerosis. They had seen the studies in rodents. They knew that there was a chance that helminths could help with multiple sclerosis, so they monitored their population, and when they found somebody who was accidentally colonized with a helminth, they put them in the study, and at the same time they took somebody who was not colonized and put them in the control arm of the study. You can see their data here that the people that got colonized, their multiple sclerosis essentially stopped progressing. And of course the progression of the multiple sclerosis was typical in patients who were uncolonized with helminths. Before this came out, there was some studies from Joel Weinstock's laboratory looking at patients with inflammatory bowel disease. Most of those patients were not responsive to pharmaceuticals. They were on their way to getting a bowel resection from a surgeon. They had not optimized the technique, but it worked very, very well. And I use the word cured for more than 70% because I view exercising the immune system with biome enrichment as a natural thing. I don't look at it like a drug. So they would say that it's treated with a drug, but I like to think of it much more as a cure because you're exercising the system, you're getting the system back the way it should be. So if you had a patient with heart disease and they fixed their diet and they got plenty of exercise and their heart disease went away, we would call them cured. So I call these patients cured in much the same way, by analogy. If you look at all the different diseases which can be affected by enriching the biome with helminths, and we're not talking about the microbiome, we're talking about other parts of the biome. On the left-hand side, you see diseases that are confirmed. By confirmed, we mean somebody ran a study either in humans and or in laboratory animals with a control group. It was blinded, it was appropriately done, so we can determine what was causal. Now, if it's highly probable or very suspicious, that means that depending, if it's gender dependent, if there's a component of autoimmunity, if there's a component of inflammation, then we can think that probably biome depletion is involved. And we have some data that address these. I'll show you later on just from some of the work that I've done in my lab in humans as well as in laboratory animals. A lot of people ask me, why doesn't everybody already know about this? How did this sneak up on us? And the answer is, if we had gotten a toilet, one day and then suddenly gotten sick the next day, then we would have known toilets are bad. But toilets are not in fact bad. They prevent infectious disease spread, and it does take a while for biome depletion to add up. Modern medicine really focuses on more minutiae, molecular genetic mechanisms, than it does on the big picture. That's how modern medicine has trended. It's tied in with how modern medicine works with the pharmaceutical industry. I've done some studies looking at the mechanisms. I think our best studies looking at how helminths will affect immune function were done in wild rats. I won an award for this. I'm very proud of that award, Worst Jobs in Science, in 2009 from Popular Science magazine. I think in the interest of time, I'm going to skip through that data. This is a very simplified slide of your immune system. Green means that we found changes. We didn't find changes where we expected to find changes. There's a laundry list. This will be on the YouTube presentation. A laundry list of different kinds of changes that, to an immunologist, all of those look pretty scary. A final slide here on why we're having problems figuring this out and using modern medicine as the basis. The reasons are, simply put, that modern medicine really doesn't tend to evaluate changes in culture that happen over a period of 40 or 50 or even 150 years. We tend to look at a single patient at a single time and try to figure out what's wrong with that patient right now and how can we get them back to normal, which essentially boils down to looking at a fish out of water, waiting for it to get sick, and then trying to fix it with chemicals. It's not necessarily the appropriate approach in this case. One problem with trying to fix these vastly complex systems with a single chemical is simply that they are too complex. There's so many interactions that are involved that trying to repair this with a single chemical or even a handful of chemicals or pharmaceuticals is very difficult. And this is just a set of a few slides showing a few different pathways that are involved in immune function. And as you can see, it just goes on and on. It's almost unimaginable that we could make complex changes to our environment, derail the system, and then hope to fix it with pharmaceuticals. We've done some experiments on this. I want to show you these because they relate to cognition in very young mice. So we're very young rats. We're basically showing how we can use a helminth or biome enrichment to protect the brain of very young rats. So Susan Poulton and John Poulton, Susan especially, did a tremendous amount of work. It was a labor of love doing this project, and it was funded by Safe Minds. I have to thank them for their for the funding for this. What we do is we take our rodents, we enrich their biome, which can mean adding in a helminth, or we can put them in a more complex, a more enriched environment if we want. Most of the data I'll show you are just adding in a single helminth. Then we breed, so the parents have their biome enriched, and we breed the animals, and in collaboration with Stacy Bilbo, what we do is we give them a 2-hit model. So very early on, at 4 days of age, which would, in terms of brain development, that's very early in humans. They get an infection with E. coli, and then later on, at 60 days of age, which is, say, a couple of years old in human years in terms of brain development, they get a small challenge with lipopolysaccharide, or LPS. It's a component of E. coli. So LPS by itself is not dangerous at all, and it should have a very minimal effect. We then assess their cognition, and we assess their immune function. So what you find, and there's a picture of Stacy there, what you find is in the green there's some decrease, but it's not significant. And by decrease, I'm looking at the ability of the rats to think. And it's the hippocampus is involved, their ability to sort of remember where they're at. And if they can remember where they're at in this test, they will freeze up. So it's as the line drops down, the rats are getting a mental fog. They're having problems with their cognition. So what you see in the biome depleted, or just normal laboratory rats, is that there's a very profound and statistically significant drop-off when they get the stimulus. But if you enrich their biome, much of that drop-off is attenuated, and there's a significant difference between the biome enriched and the biome depleted rats when you give them the stimulus. I think what's a little bit more shocking is that you can measure changes in the brain on day 4 of these animals when they get the infection. So the green bars show what happens on the left-hand green bar, the lighter green bar. You've got a normal rat that's 4 days old. You're looking in the brain at the hippocampus at immune cell activation. And what you can see is it's a little bit higher than the control. And we don't really understand why that is. The rats are fine. They're cognitively normal, but there are differences in their brain. What happens is when the animal gets an infection, not in the brain, but gets an infection down near the gut region, if the animal's biome enriched, something changes in the brain and there's less immune activation. So you get an infection in the gut region, and inflammation in the brain, or markers of inflammation drop. We don't think the controls are inflamed to start with. They just have a different baseline. The problem is that if you're biome depleted in the gray bars, when you go from the left to the right gray bar, you get a profound increase in inflammatory markers in the brain. That increase then is associated with what we showed on the previous slide, is kind of a lifelong sensitivity later on. When you get another stimulus, then you can have cognitive dysfunction. So it's an early exposure model showing that early exposure is having an effect later down the road. Stacy does run the controls showing that if you don't get that early exposure, then the brain is not sensitive. So we've also looked at the microbiome on this. If you add a single helminth to the microbiome, it will change, or I'm sorry, if you add a single helminth to the gut, it will change the microbiome. So on the left-hand side, you're looking at animals with no helminths, and you see a lot of those red bars. Each different color bar represents a different operational taxonomic unit or group of species that we talked about earlier. On the right-hand set of bars, we've added in a helminth, and you see the red bars go away. The red bars are a bacilli, which is bad for, it's associated with the Western diet. It's probably not a good thing to have a lot of in your microbiome. Those are gone and replaced by a Clostridia species, or groups of species, which tend to be protective against allergy. And Aaron McKinney is the one that did the work. On the right here is kind of a breakdown of how we're seeing a shift from in the green to the bacilli, to the red to the Clostridia type species. So I want to spend a little bit of time on this study. We're very excited about this study. It is just coming out in the next couple of weeks. You won't be able to find it online yet. Again, it will be a couple of weeks. It has been accepted for publication. It's in it's in press right now, which means the production editors are working on it to get it typeset and so forth. So in this study, we're looking at people who and we're collecting surveys from and interviewing people who are treating themselves with helminths to see how the helminths work. So we have 3 different sources of information. One of those is just what's publicly available from websites and books and blogs. Another one is actually interviewing the people that are producing and providing the helminths, and then we did send out surveys for people who were using helminths to describe for us in great detail how the helminths were affecting their medical conditions. The studies were performed by Anna Chang. She's the one that collected almost 200 of the observations from the publicly available information. This was done in collaboration with Janet Wilson. She's a professor of sociology at the University of Central Arkansas, a longtime friend of mine, and we're very happy to have her helping us with this, what is basically a sociological study as much as it is a medical study. And the reference is on here. Again, that will be on the YouTube. Again, you can't find it right now. The first thing I'd like to point out is that there are several places that people are getting their helminths from. And of course, we don't recommend that anybody try this, and certainly we don't recommend that anybody don't try this either. We can't recommend anything from a medical perspective because the controlled trials have not yet been done. Tanawisa is the company that's been around the longest, even though they haven't had that name for such a long time. They've been around about 10 years now. The cost is, it can be more than $10,000 per year per person, so many people can't afford that. But keeping pigs and isolating whipworms from the feces of the pigs is an expensive task. There's a couple of more companies that most people are aware of, autoimmune therapies and worm therapies, that popped up about 8 years ago, shortly after Tanawisa came online. There's 2 much more recent companies, Biome Restoration and Wormswell, and you can see over on the right-hand side they're selling different species. I'll talk a little bit about those species in a minute. I put Biome on here. They sell soil bacteria. It's a way of biome enrichment. They don't sell a helminth. It's the bacteria that most of us are missing that we would have normally been exposed to constantly before the Industrial Revolution. They're ammonia oxidizing bacteria. This is all described in the paper. This is our estimate, mostly based on interviews with providers. We talked with a number of commercial providers, a couple of providers that are not commercial. There's a couple of community providers, people just with social networks that raise helminths and distribute them among their friends and social connections. Based on all the numbers we've got, most people are using the porcine whipworm. There's probably about 6,500 people or so, between 6,000 and 7,000 people that are using helminths in the world today. When we collected our surveys, we had a very strong bias. And now the next slide will show you, if you take a look at these numbers, sort of take the layout here, you'll see in the next slide that when we collected the surveys, we saw we got a lot of returns of people using the rat tapeworm. There's the survey numbers, and the reason why is because one of the providers who's non-commercial decided to help us. We're very thankful for that, and that provider had 100% return rate on asking people to fill out surveys, whereas most providers warned us it's very hard to get people to fill out surveys. When they're paying for their helminths, they don't want to fill out a survey. I think with the porcine whipworm, we had some problems with the demographic there. Those people might just be hard for us to reach, since a lot of the way we reach people is via social networks. So the human hookworm surveys, we can get those more readily through the social networks. The important point here is that we've essentially eliminated survivor bias, a kind of bias in these surveys from only people who are, most people dropping out. and then only the remaining people filling out the survey. So we eliminated survivor bias with the rat tapeworm. It's very convenient, helpful to us, but again, the bias is towards the rat tapeworm. I'll let you look at this slide just for a minute. If you ask people how to rate the effectiveness of their helminthic therapy compared to the effectiveness of whatever medical treatments they've tried, the helminthic therapy does very well, as you can see. But when we look at the next couple of slides, it's important to note that people that are filling out these surveys are comparing their helminthic therapy. In the back of their mind, they're comparing how their helminthic therapy works with whatever pharmaceuticals that they've tried. So the first slide, this was surprising to us, honestly. We published a paper that predicted that helminths would have an effect on cognitive function. We published several papers, but we were sort of surprised to see how beneficial the effect was, especially trying to reverse something that has already taken place. So each individual gets 2 numbers. So you look up at the anxiety disorders. We had 5 respondents with anxiety disorders. The first number is the effectiveness, and then there's a slash, then the second number is the side effects. So a 9-0 means it was very effective with absolutely no side effects. They're scoring things on an 11-point scale. This is the 11-point scale here from 0 to 10. So you can see it's relatively effective. If it's bolded and underlined, that means based on the information we have, we know that the person was underdosed. In fact, the same individual had both anxiety and depression, so gave the same score on both. You can see the 6.5/0, and that individual did increase their dose. The supplier just told me what had happened, and they are now, as far as I am informed, completely free of anxiety and depression. So these are a couple of more tidbits of data. I've got a number of anecdotes from suppliers as well. These are, again, this is from survey data showing that the helminths work pretty well on migraines. Again, this is very biased towards the rat tapeworm, since that's what most of my data from the surveys are derived from, people using the rat tapeworm. I'll put those references up there. They'll be on YouTube. The point is that people have been predicting that helminths would affect cognitive dysfunction. If you want some general reading, I recommend this article. It's a great article written by Emily Deans in Psychology Today, The Parasite Ate Your Depression. A wonderful article. I think it's easy to understand. Most people think that if you can alleviate depression and allergy and autoimmune disease, that the therapy must be immunosuppressive. Cindy Pye, a student in my laboratory, did a tremendous amount of work enriching the biome of laboratory rats and showed that when you enrich their biome, their immune responses actually get better. So you can see in the green when we inject a bunch of a very complicated antigen cocktail into some rats, the cocktail elicits a much better response, and then in other words, the immune response is better in the laboratory animals with biomes enriched compared to normal laboratory animals, which are biome depleted animals. Our normal laboratory animals don't have a very rich microbiome. They certainly don't have any kind of helminth in them, because we screen those out and we make sure and we treat our rats so that they don't have those. I'm going to address a few issues about helminthic therapy now. In this slide, in the big green circle, I'm showing a couple of horrible organisms that will train the immune system. I think they're the amoebic dysentery, the porcine tapeworm, which forms cysts in the brain, and the organism that causes malaria are things that would prevent allergies most likely, but we're really not interested in those. We're going to weed those out and select the ones that we want, which are essentially benign in terms of causing problems. And if you have questions about what kind of side effects people have with helminthic therapy, I can talk a little bit about that in the Q&A session, and certainly that paper is coming out, not the paper on this slide, but the paper talking about self-treatment. It's a very thorough paper. It's almost 17,000 words long. So tremendous amount of detail in that paper, and you're welcome to email me if you have questions that aren't answered in that paper. Back to our domestication process, the idea is fairly simple. People are already starting on phase 1, which is just to look around, try to find the helminth that treats disease but doesn't cause adverse side effects. We expect over the long term we'll go into a second phase, which will involve engineering, trying to breed or select, in some way devise helminths which are long-lived, easy to produce, very low side effects, maybe designer helminths for certain genotypes. We know that different populations, African versus Norwegian, for example, may be adapted to different kinds of helminths. So there's a lot of work to be done. We're only at the very, very beginning stages of it. Only about 5 helminths have ever even been tried for helminthic therapy. There's dozens of possibilities. So a lot of people ask me about marketing of helminths. How are you going to get people to swallow a worm? Well, it is in the marketing. There's no question about that. On the left is an electron micrograph of a human hookworm, which has become a very famous electron micrograph. It even looks like it has eyes, which of course hookworms do not have. On the right-hand side, you can see it's called a HDC, or it's a sister cercoid. It's a certain stage of the rat tapeworm. It's the therapeutic stage for humans. And inside there, most people think they can see the face of an angel. That's not a very scientific observation. I don't know that the face of an angel has been recorded, but I'm just saying this is what people are telling me. The last student that I had that absolutely protested about this, we were at lunch. I had just given a seminar, and it turns out that she was eating a can of germs at the time. So I pointed this out, and the idea being that marketing can be very effective, and it can, it's all about the marketing. So I don't think that's going to be a problem, especially if we know what kinds of diseases that that can be prevented or treated with helminthic therapy. Right. I'm going to skip a couple of slides here and go down to the prediction that vaccines and antibiotics are things we use so that we can live in high population densities without dying from infectious disease, whereas I think biome enrichment or biome reconstitution is going to be something that we will need so that we can live in a healthy way in high population densities. We need to take a step back and make sure that we don't think of biome enrichment as a kind of drug. We don't think of helminths as a drug, but as sort of part of a healthy lifestyle. There's other things that do hammer on our immune system. I believe there is one tribe or one group down in South America that they are fairly sick with inflammatory disease, and it's because they've imported the Western diet. They've started popping antibiotics, which affect the microbiome, like their candy. They're not regulated down there. They still have helminths, but they are also getting sick. So there's other, it's not, the helminth is not the magic bullet. It does have a profound effect, but at the same time, there's other factors that we need to consider and look at health overall. We can tell from our survey that most people who are using helminths are not even checking their vitamin D levels. They don't even know what they are. So we humans do have a tendency to focus in on one thing, gravitate toward that thing as the magic cure-all, and that's really not what we're looking for here. And at Duke, we are going to start clinical trials looking at helminths with cognition. So we're sort of guilty of doing what I'm saying we shouldn't be doing, but the idea is to look at biome enrichment as part of a healthy lifestyle, not as a kind of drug to fix things. I'll skip a few slides just to remind you here the model we have for autism and how it develops. Oxidation is probably a big issue. Biome depletion is a big issue. You could think about a lot of other things, though, that affect the immune system. This is a paper we've just published, or, well, it's about to be published. It's accepted for publication, looking at chronic stress. And there's a vicious cycle with chronic stress. And, of course, if you have children with autism, that's known to be very stressful. It does cause problems, and chronic stress does cause immune dysfunction. So if you want that paper, will be available soon. It will be publicly available free of charge. You'll be able to look in detail at the kinds of things in our modern society which cause stress. And I think this will be my last slide before the credit slide. It's been known for a long time that the adult brain is affected by all of these factors, and I think if we imagine how these factors, from psychological stressors to poor diet to vitamin D deficiency affect the adult brain, it's not scientific, but it's very reasonable to expect that they'll have a tremendous impact on the fetal brain during development. I think that's something that we really have to consider when looking at all of the cognitive dysfunction and the psychological disorders that we find in the youth today. I've got a series of slides here comparing our human body to an ecosystem. You can see how we've affected the ecosystem here pretty profoundly. A lot of changes there. We've changed our interaction with food. So we don't have to work to get food. Instead, we have a rat race or a paper chase now that most of us engage in that causes a lot of dysfunction and chronic disease. The references are here. We'll put the references out so that you all can find those when you need them. And I'll end with this acknowledgement slide. I've worked with a lot of people who have expertise outside my own area. Again, we'd like to thank Safe Minds for their work. Shu S. Lin is a collaborator. It's been a very dear friend of mine and a great supporter for many years now. Zoe Holzknecht, Mary Lou Everett, and Susanna Mesa-Coyton have run the lab and taken care of everything so that I can be the flaky professor and not have to worry about how things actually function. And I'm just, without these folks here, nothing that we're doing now would have been possible. So I'll leave it on this slide here, which is showing a couple of the things that we need to do in the optimal therapeutic zone. We don't want to take away toilets. We don't want to take away our indoor work environments, our air conditioners, and our soap and our detergent. But we do want to deal with the consequences before we get down to the bottom part of this graph where you have lots of vicious cycles. I've just put in biome depletion and vitamin D deficiency here, but I could put in chronic psychological stress and inflammatory diets as well as a sedentary lifestyle as well. So at this point, I think I'll take a pause and take questions.
Okay, thank you. That was a great presentation. We've got a lot of questions, so I want to let people know if they're still on the line that you can type in your questions and we'll try to get to as many as possible. The first one.
Could I say first that I'm used to speaking with the audience that I can see, so I can tell when I've said something confusing. So I apologize, I can't see you. I'm sure I said some things that may not have been very clear, and I'm very happy to receive the questions.
Okay, great. The first question, they're asking about Helminth therapy, and they're asking about is there a link between the effectiveness and the price? So they're asking, do you get what you pay for, or is there any evidence that there's superiority of different products?
Well, so the price is a complicated. So the porcine whipworm is pretty easy. That's the very expensive one, and the reason why that is priced so high is because you have to have a huge production facility, and you have to keep clean pigs. The price did recently drop by 40% because they changed their production method and made it much more efficient. But it's still by far the most expensive helminth, and it depends on how much you need. With all of these helminths, the general idea is you start with a certain dose and you kind of ramp them up until your symptoms are relieved or until you start getting symptoms, side effects that are not tolerable. The porcine whipworm is very effective. The only complaints about it that I've heard of are the cost. It's out of range of most Americans. So we don't have as much. We have some data about the rat tapeworm, and so different organisms affect the human body different ways. So with those cognitive effects, we can't be absolutely certain, but it may be that the hookworm, that's one of the worms that colonizes the body and stays in there for over a year often, doesn't have the same type of effect on cognition as say the rat tapeworm or the porcine whipworm. And interestingly enough, the porcine whipworm might have a better effect on cognition, and it's based on what we can tell. And it's hard for us from the data that we have to compare different helminths, because most people don't flip back and forth from different helminths and then write a comparative article about it. They get with one supplier and they tend to stick with one supplier. So we're speculating on this, but it looks like the porcine whipworm probably works better than the human whipworm, at least on systemic effects and having effects on cognition. There's fundamental differences in the way those two worms work. With the porcine whipworm, you have to get dosed every 10 to 14 days. So you're constantly getting exposed to an organism which doesn't last very long, whereas with a human whipworm, you're getting exposed to an organism that can last up to 5 years in the human body, which is longer than anybody thought before people began self-treating with helminths. So I know that's a vague answer. Bottom line is probably not, probably no. The cost is not directly proportional to the effect. It has more to do with the production and what it takes to produce it and distribute it than anything else. The rat tapeworm has only been on the market for less than a year now, and it has shot up in popularity beyond the human whipworm. If you recall the data, despite the fact the human whipworm's been on the market for 8 years. So things are in flux, things are changing. Though as people try new helminths, things will continue to change.
Okay, so the next question, this is a great one. They're asking, aside from helminth therapy, what else can we do to improve biome depletion?
I think one of the best things you can do is when you have to take antibiotics, make sure you take probiotics. That will help, because so you can't do very much to protect your helminths, and because we live in our society, the helminths are essentially extinct in this part of the country. And if you did try to get helminths, you'd probably end up getting something much worse than a helminth, and you wouldn't be happy anyway. So the helminths are sort of, we have to artificially reintroduce those, but I think we can protect our microbiome. And the microbiome is obviously extremely important. Vitamin D supplementation will help you protect your microbiome because it helps your immune system interact effectively with the microbiome. Taking probiotics when you have to take antibiotics is very helpful. And the diet is incredibly helpful. In Western inflammatory diet, high in processed sugar, low in nutrients, high in fat, it's bad for your microbiome. It doesn't give it any prebiotics or fiber inside, soluble, insoluble fiber. It doesn't give the microbiome the thing it needs to really thrive and become diverse. So yeah, that is a great question.
The next question is about, have you done any research on fecal transplants, and do you have any insight about that process?
Well, I've worked, so fecal transplant is it's a simple thing. The clinical data show very conclusively that if you have recurrent C. difficile colitis, it's a specific kind of colitis or inflammation of the gut that you get following treatment with antibiotics. It's associated with a certain organism, Clostridium difficile, which is different than the Clostridia that are protective against allergies. It works great for that, and if you're, obviously if somebody's at a institution which doesn't offer a fecal transplant for recurrent Clostridium difficile colitis, then they probably need to go find some place that does offer that procedure. The FDA does allow it to happen. It's an easy procedure to undergo. Now, a lot of people are trying fecal microbiome transplant for a lot of different things, and sometimes, it's hit or miss, and I think the clinical data are not looking good for that, just in terms of treating inflammatory bowel disease. And I think the problem is because a lot of these inflammatory diseases of the bowel, they involve immune system dysfunction. So just by replacing the microbiome, you don't necessarily fix your immune system. Again, very hit or miss. The clinical data right now don't support doing the fecal microbiome transplant for anything other than recurrent C. difficile colitis. And that's not to say that some people don't receive incredible benefit from that. It's just that the clinical data are not supporting that at the present time. And clinical data are dependent on a lot of things: how the patients are selected, how the transplant is done, although that doesn't seem to be a big deal, but patient selection is probably a big deal. Did I dodge that question okay?
Well, you mentioned probiotics earlier.
Yes.
People are asking, should we take probiotics all the time or just with antibiotics? What's the relationship there?
The clinical data show that, and by clinical data I mean somebody did a trial, somebody bothered to do a study and had a control group and looked, and does it make a difference in a blinded way? And the answer is that when you're taking antibiotics, we know that the microbiome is at risk, and that's why you can get recurrent C. difficile colitis. And yes, the probiotics, particular probiotics that have been tested, do effectively help protect the microbiome. A lot of people experience great benefit from routine use of probiotics. There's been some studies done in animals showing that probiotics can protect the gut from, if they're given in advance, can protect the gut from, say, some kind of infectious problem. So the answer is we would never recommend against people using probiotics, but there's not a lot that we can recommend in terms of this really needs to be done because the clinical trials have been run.
Okay, so here's a question about measuring general gut flora. They're asking, is there a test that can be done to determine which areas are weak and which are strong?
Well, of course, as I showed some data that from our laboratory rats, it's easy to take those kinds of big, broad measurements in humans. You may have some questions about what does the microbiome look like in patients with autism. There's some great work being done out of Arizona showing that I think it shows. I think the data are very good, showing that the microbiome in patients with autism is almost like a hyper-westernized, and that could be part of the diet and patients with autism having a poor diet. But you can look at the microbiome, but then using these kinds of techniques and get vast amounts of information. You can get 100,000 different sequences for very low cost right now, but it's not real clear what that's telling you. It goes back to an earlier comment I made. How do you know that the microbiome is really the problem versus you've got some kind of immune dysfunction? There are some tests that some doctors are running looking at the amount of secretory IgA and other kinds of inflammatory markers in the stool samples. I think those can be helpful, but again, there's not been a lot done in mainstream medicine looking at those. They're not currently used in mainstream medicine as diagnostic tools.
Okay, so you also just touched on diet, and I've got a lot of questions about what kind of recommendations specifically. Do you have specific diets that you've seen people succeed with if they're trying to recover their microbiome, or does it just really vary by organism, essentially?
Well, first, it's important for me to throw in a disclaimer that all of my patients weigh about 350 to 500 grams, and they have fur, not hair, and they have a tail. So my patients, they're on a pretty good diet for the most part. Now, I'm not a dietitian, but of course we know that in general, pizza is bad, and grilled vegetables with some fish is good. So we know what an inflammatory diet looks like. It's a lot of processed sugars. It's a lot of processed carbs with very low nutrient value, and it's high in fat. We know that before the agricultural area, wild game has an average of about 5% body fat, whereas all our domestic animals now are approaching 20% body fat. Plus, then we purify the fat out of things so that we can, get even more fat, corn oil, for example. So we know that less fat and less processed carbs are good. Again, I'm not a dietitian, so dissecting out all the details of that, that's something I would leave to a dietitian.
Okay, great. So one person's asking here if you're planning to repeat the study on cognition with various types of whipworms to determine if there are differences.
So I think that's a great question, and it relates to another very interesting question. If we've discovered 10 years ago and 8 years ago, respectively, that helminths will prevent inflammatory bowel disease or treat inflammatory bowel disease and very effectively treat multiple sclerosis, why haven't people been looking at this? And the simple answer is, being at a medical center, we're looking for just a way to do the study. So those very early studies, it wasn't so easy to follow up on those. You can see, the very first studies were done with a porcine whipworm, and you can see how much it cost, to come out eventually with that product. So if you did a study with a porcine whipworm and it worked great, then when you stop the study, then the patients can't afford the product. What are you going to do? And you can't, if you're a clinician in a medical center interested in a less expensive helminth, say a human hookworm, you can't get your institutional review board, your IRB, you can't get those guys to approve you going down and trying to pick up some random, some worm from South America. So right now we are focusing on what we can reasonably do that we think that, number one, we can produce the helminth. We have to be able to produce the helminth. We have to be able to produce it at a reasonable cost, and it has to be something that's fairly benign. So it's going to be tough, and it has proven tough. I think this new helminth that's on the market now is something that I produce in my own lab. A lot of laboratories have produced it for many decades, and nobody ever thought to use it in humans. So we can use that helminth, but then when you talk about getting other helminths and comparing them side by side, I think that's more down the road. I think the first steps are just to run the controls, do the blinded studies, see can we either prove or disprove that this really does what we've always thought it would do and what it looks like it is doing in these patients that are self-treating. So that's where we're at right now. So down the road, yes. Down the road, I expect that companies will get involved. There will be designer helminths. I think I showed the slide showing the different stages of domestication, and people will be seriously looking at which helminths for which condition, and how do different helminths compare. It's important to note that it depends on how you prepare your helminth. So with the rat tapeworm, for example, there's 5 or 6 things you can do to that worm and cause it not to work very well. And it depends on how you feed the host that it lives in. It depends on what temperature it's stored at. It depends on a number. It depends on how old it is. If it's too old, if it's too young, it doesn't work very well. So those are really what we're interested in first, is just seeing if we can get a single helminth to work in a rigorous, defined way. I think that's the problem they had with the clinical trials with the porcine whipworm. There's a lot of discussion about that in that helminth self-treatment paper that's coming out soon. I'll stop there on that question.